Hi everyone, welcome to Useful Genetics. This is the overview video for the first module. And in this module, we're going to begin and end by thinking about the genetic differences between different people. This is going to then lead us to thinking about the basic processes of molecular biology. We'll review all of the basics of DNA biology, transcription and translation. Most of you will be at least partly familiar with these concepts, but don't panic if you're not. We'll review the basic molecules, and then we're going to connect the molecules and the processes with the ideas about the storage and flow of information, because genetics is as much an information science as it is a molecular science. We'll think about the natural diversity in populations and the evolutionary histories of these populations. And then we'll bring it back to think about the evolution of the human genetic differences that we see. Of course, all of these topics are interconnected, and each of them will help you understand the others. Now, there's a long list of lectures, but the lectures are quite short. For many of you, if you've recently taken, for instance, a college introductory biology course, a number of this material will be somewhat familiar, depending on how much you forgot since the final exam. So, for example, most of this basic molecular biology will have been covered in an introductory biology course, as will simple information about human life cycles. But in addition to refreshing your memory about this, I think you'll find that it also is providing a lot of new perspectives and new context on this material. So even if you're fairly confident in your basic understanding of these principles, it's well worth your while to watch all the videos. Now, coming up next is Lecture 1A, where we're going to start thinking about how different are we. In particular, we're going to introduce the very important concepts of phenotype and genotype. I hope to see you there. Hi everyone, I'm Dr. Redfield. Welcome to the very first lecture of the first module of Useful Genetics. This lecture has an abbreviated version of the structure that you'll see in the whole module, in that we'll start and end by thinking about human genetic differences, and in the middle we're going to introduce some very basic concepts, in this case just the very first introduction to DNA. Now, as people, we're really good at noticing differences in appearance of other people. That's probably because we're social animals. We're also really good at noticing the ways in which people from any particular part of the world look different from us, but very much like each other. And it's often said, well, we're all the same under the skin, but are we really? This is a question that genetics can answer. To think about this, we need to introduce two very important terms that will come up again and again and again, and that's phenotype and genotype. These terms were invented by the very first geneticists as a way of distinguishing between two things. First, the phenotype was the observable properties of organisms, what you could see, the features that we cared about. And it's come to include not just what you can see with your eyes, but the observable, the molecular properties, behavioral properties, anything about an organism. When the term genotype was invented, it was because of the brave inference that these phenotypic differences that we see were caused by differences in some mysterious entities which were given the name genes. Different versions of genes were the different genotypes. We now know that this was a correct inference, and in fact, that the different versions of genes are in fact different versions of DNA sequences of chromosomes. Now, quick refresher of information about DNA. All of our genetic material is organized into long molecules called chromosomes. And these long molecules are chains of subunits called bases. So it's a long chain. And in the DNA and other what are called informational molecules, the bases aren't all the same. Instead, they come in 4 slightly different versions, 4 slightly different subunits, called by the names A, G, T, and C, which are just the single letter abbreviations of their chemical names. We often represent DNA as the order of the bases in the sequence because, in fact, this order of the sequences is the genetic information. It is the differences that cause the phenotypic differences that we see. So, if we think about DNA in the life cycle, we know that each egg and each sperm contains one complete set of DNA molecules, 23 chromosomes. And when the egg and the sperm meet, when the sperm fuses with the egg, the resulting fertilized egg contains 2 complete sets of genes, one from each parent. Individuals develop because the fertilized egg undergoes many, many cycles of growth and cell division, producing all of the cells in our body. And most importantly, from a genetic perspective, all the cells in our body, for instance, every cell in our arm, all our cells, contain the same 2 sets of genes that were in the original egg and sperm. Now, what is this complete set of DNA molecules? Well, as I said, it's 23 long chains called chromosomes. The other very important information is, well, how many subunits are we talking about? We're talking about 3 billion subunits, 3 followed by 9 zeros. Now, we speak of this complete set of DNA molecules as being a genome. And we can use the word genome in 2 different senses. The first is we can speak of the human genome, by which we would mean a reference sequence, like might be stored in a computer. But really, when we think about humans, we need to think about the genomes of all humans. And the genetic diversity between us. Together, this makes up the real genetic endowment of all humans. Now, how different then are our DNA sequences? Well, from one perspective, they're very, very similar. So on average, they're 99.9% identical. A way to visualize that is to consider the set of DNA that you inherited from your mom and the set that you inherited from your dad, shown here in blue. Here I'm just showing 1,000 bases. If you compare them, what you'll find is that on average, in every 1,000 bases, there's just 1 sequence difference. Now, what if we were to compare a set of DNA from you and from one of your neighbors? Again, we pick 1,000 bases and compare you and your neighbor. And what we'll find is, well, on average, there's 1 DNA sequence difference. Even if we were to compare 2 sets from people from other sides of the world, again, we'll see on average there's just 1 difference. So it doesn't make any difference really how far away we come from. We're all very similar. But as I said, there's 2 perspectives. And the other perspective is to think about, well, how different are we? So I'm going to let you calculate this in the first of what are many self-test questions. And the format will always be, there's a slide here where I'm introducing the question to you, asking you to calculate how many genetic differences are there between you and your neighbor. And on the next slide, you'll see Coursera's version of this question. And after you've done the arithmetic, you can check your answer. And then finally, we'll come back to a slide where I'll discuss the answer. Here's the Coursera question. hope you got that right. Right answer is there's 6 million differences, a lot of differences between you and your neighbor, between each of us and each other one of us. Where did this number come from? Here's the calculation. There's 1 difference per 1,000 bases. There are 3 million bases per set. And there's 2 sets per person. So that gives us 6 million differences. So how much difference do these differences make? Well, that's kind of a silly question. If we put it genetically, we will ask how much phenotypic difference do these genotypic differences make? And the answer is, well, most of these differences don't make any detectable difference to phenotype. And that's because most of these differences are in parts of the genome that are really not important for determining our phenotypic properties at all. We'll talk about this a lot more in a later lecture. But the rest of the differences cause all of the heritable differences between people. And just in case you're thinking that people are kind of special, we're not. Exactly the same situation exists for all of the other organisms on the planet. Their sequences of any one species are very similar, but there are so many bases that there's still a lot of differences which cause all the phenotypic differences. Now, what we've done, we've introduced some new words, especially phenotype, genotype, and genome. We've talked about the genome in the life cycle, and we've talked about the differences between the genomes of different people, both how similar we are and how very, very many differences there are. Now, coming up next, we're going to talk in more depth about the properties of DNA. And following that, we've got a series more lectures in Module 1 talking about the properties of genes, of chromosomes, of life cycles, and finally, how we can compare DNA sequences to learn about human history. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is Lecture 1B, where we're going to introduce the properties of DNA, thinking both about DNA as a physical molecule with physical properties and as an informational molecule, how these physical properties allow DNA to be the carrier of genetic information. Now, DNA is both. It's a physical molecule and it's the genetic instructions that are used to build our bodies and the instructions that are passed on to future generations. How does this work? Well, first, DNA is the kind of molecule that's called a polymer. As we said earlier, it's a chain of identical or nearly identical subunits. In DNA's case, as an informational polymer, the subunits need to be different because it's the order of the subunits that creates the information. In DNA, there are 4 kinds of the subunits called bases, and they go by the names A, G, C, and T. DNA typically has 2 strands, so we talk about it as if it was one molecule, but really the functional DNA in a cell is 2 molecules wound around each other. We often draw it like this to indicate that. And in these 2 strands, the bases that are across from each other are physically complementary. They're not identical, but they fit together. And the meaning of that is shown here. So this is just a schematic representation of the 4 bases as 4 different shapes attached to a line that represents the DNA strand. And the important feature about these 4 shapes is that they are pairwise complementary. So the shape of the A base fits with the shape of the T base. The shape of the G base fits with the shape of the C base. But they don't fit in other combinations. If we try to pair an A with a C or a C with a T, either they don't fit or they bump together. Now, this fit is mediated both by complementary shapes and by complementary charges, the mixtures of pluses and minus charges on different parts of the base molecules. Now, these interactions of shapes and charges together create a kind of chemical interaction called hydrogen bonding. These are individually weak but collectively strong chemical bonds that serve several very important physical functions for DNA. They hold the DNA strands together so they don't come apart. They direct DNA replication so that a new strand can be synthesized using the old strand, and they direct the synthesis of the related molecule called RNA. Now, these sequences, these base pairings, then also create the genetic information in the DNA, the order of the bases along the chain, because that's what specifies protein sequences, which are proteins are the workhorses of the cell, and it also specifies other genetic functions. So the two strands have complementary shapes and charges. Thus, the two strands have complementary information as well, the information being the sequence of the bases. Because of this, it's possible to do error correction. If one strand is wrong, the other strand, the bases in the other strand, provide the information to correct the mistakes. This is called informational redundancy. And it's a very important feature of all informational systems, including the computers that I'm recording this on and that you're watching this on. It also allows for copying of the information, duplicating of the genetic information for heredity, and it allows for readout of the information to instruct the cells to produce proteins. Now, here's a drawing of DNA, just a schematic drawing. As I said earlier, the DNA in each chromosome is two very long single strands, and you can follow these in this chemical structure along this backbone of each strand. And these bonds that hold the strands together between one subunit and the next subunit, these are strong bonds. They're not going to come apart at all. In contrast, the bases, and here are the bases, A and T, C and G, held together. They're held together by hydrogen bonds that, as I said, are weak bonds. So they're fairly easily pulled apart. Individually, they're weak. In the whole molecule, summed up over all the bonds in the molecule, they're very strong, holding the DNA together, but it's relatively easy to pry individual bonds apart. This is important because the strands have to separate when the DNA replicates or when the bases are transcribed into RNA to direct protein synthesis. One other point I didn't make is that these molecules are asymmetric. So if you look at it from this side, it's different than if you look at it from this side. They're like text. So the letters in our alphabet are asymmetric. If you write them backwards, they mean something different. And the text that we read, the text on this screen, for instance, has to be read in a particular direction to make sense. In Western languages, we read from left to right. In the same way, the DNA strands are asymmetric, and they're asymmetric in a particular way. We can think of each strand as having a direction, and we refer to the ends by numbers, 3' and 5', and we would put an arrow at the 5' end. So the other strand is running in the other direction, from its 3' end to its 5' end. Now, one final feature of the double helix, when the two DNA strands are wound around each other, and that is that even though the bases are interacting with each other in base pairs, the sides of the bases are still exposed on the side of the double helix, like the sides of a rope. so that it's possible, just by looking at the sides of the DNA, for a protein to sense which bases are present in which order. And the importance of this will be seen in this slide here. So there are two ways that the physical properties of DNA become information. And the one that has to happen first isn't coding for proteins. Instead, it's the ability of specialized DNA binding proteins to feel the shapes of the DNA and bind to particular sequences. And that's illustrated here. Here's a DNA molecule, a regulatory protein feeling its way along the DNA until it comes to a place in the DNA that has the right sequence of bases that it's able to bind to. So that would be these base pairs here. This protein can feel the sequence of the bases, even though the DNA is still base paired in its double helix. And the big green arrow here indicates that this protein, having found its appropriate sequence, is going to cause something to happen. Maybe DNA replication, maybe some other process. Now, the base sequences of other segments code for protein. And the way they do that is that they undergo processes called transcription and translation into polymers, other polymers, the informational polymer RNA, which we'll describe in the next lecture or the one after, and protein. We'll talk a lot about protein in Module 3. The DNA sequence specifies the sequence of these molecules. This is just a little diagram of the process of transcription, where we have double helical, double-stranded DNA that's come apart. It's unzipped so that this protein can make an RNA copy of one strand. So what have we done? We've talked about DNA's physical properties, how it's made up of two strands, each with a strong backbone, and with base pairing between them that's highly specific between particular pairs of bases, A with T, G with C, and how the sides of the bases are exposed. These physical properties then make possible the informational properties, that there are sites where regulatory proteins can act, and base pairing allows for accurate DNA replication, and then, as we'll see, allows for specifying the sequences of the proteins that are to be synthesized. Now, coming up next, we're going to take just a few minutes to talk about how we represent DNA, because this is an important issue to get straight before we go on. I hope to see you there.